Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy in my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out, and I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest. It's one thing to listen to doom and gloom about food and fertilizer shortages, skyrocketing prices, the cost of living, or your job being outsourced overseas or eliminated due to automation. It's quite another thing to hear practical, immediately actionable advice from experts who can help you reduce the fear, anxiety, and burden of these problems. Tune in now to the Surviving Hard Times podcast from the Finding Genius Foundation with Richard Jacobs.
Hello, this is Richard Jacobs with the Surviving Hard Times podcast. I'm talking with Todd Callaway, associate professor at University of Georgia, part of the animal and dairy science area. We're going to talk about microbiome and animal and dairy science related issues. So thanks for coming, Todd.
Sure, it's my pleasure.
If you would, tell me about your background and how you got into the area you're in right now.
Okay, well, like I tell a lot of my students, it's, everybody's journey is always interesting, but to wind up in ag, you've really kind of got to have a love for the area. So I grew up on a horse and small cattle farm. We had some ruins of my grandfather's dairy farm, and we bred horses and cattle and all that while I was growing up. And basically, I wound up holding the mares while we were breeding horses. And by the time I was 15, I had all the bones in both hands broken at one point or other. So it was kind of an interesting way to grow up, but you learn a lot about doing different things and some of how things interconnect. But like 90% of the kids that grow up on farm, I wanted to go be a veterinarian after I graduated college. So I started working for a vet clinic and starting at, I think, about 14 or so. And during that whole time, I learned what it was like to be a vet, a lot of the things that go into being a vet. And I was fortunate to get to work in a large animal, small animal clinic. So I got to see a lot of what's going on the farm. And so I intended to go be a vet. And I came to undergrad at Georgia. It was a great place to come to school because it was near where I grew up, but far enough away where you wouldn't have the pop-in visits from family members and such. But during my time as an undergrad, I started looking around and seeing how the gut of the animal, especially in cattle, how that plays such a role in nutrition of the animal. Because the part that really just blew my mind was when you feed a cow, you're not just feeding the cow, you're feeding the microbes, and those microbes are then actually feeding the cow. So it's this true form of symbiosis that you really don't see anywhere else to this degree in nature very often, at least. And that fascinated me that, we can have our economic impact, sustainability, health, all these impacts. Everything we want to do is mediated in cattle by this microbial population. Then you started looking in pigs and humans, and you get this hindgut fermentation. That's the same sort of microbial host interactions. And it was really cool area to me. It wasn't this hot, sexy area for research at the time. It was just kind of fundamental research. And I kind of always joke with people that the basic scientists say I'm too applied, the applied scientists say I'm too basic in what we do. And, we just kind of fit in that weird niche in the environment.
So what is it? Are you studying the the microbial digestion of various farm animals, or what's your focus now?
My focus now is twofold. I focus on trying to understand how cattle and other food animals, but cattle are kind of my main home, how they degrade feedstuffs, and how can we take that fermentation that happens and make it more efficient, where the animal produces fewer end products that aren't beneficial to the animal and aren't wasted. And part of that is things like methane production or CO2 production. Those, being able to reduce those in cattle is a win-win. And that's something that the industry has seen for years, that if you can reduce methane production or you can reduce CO2 production out of this fermentation process, then by doing that, you're improving profitability because you're not wasting food that you're giving to the animal, but you're also not impacting the environment through greenhouse gases. So it's one of those areas where sustainability and profitability align nearly perfectly. And the other facet of most of my research is looking at food safety. How can we harness this microbial population in the gut of food animals to keep pathogens out, keep them from reaching our food supply? So it's kind of two very different areas, but yet when you look at the Venn diagram of what they do, there's a lot of overlap.
It's weird because the soil itself from which we feed has its own microbiome, and that needs to be cared for. So it's like the first stomach of everyone, it feels like.
Yeah, it is. And then the plants have their own microbiome. The irrigation water or even rainwater in some cases has microbiome in it. And everything is a mixture of these microbes from soil, environment, dust, migratory birds. Everything comes together. So it's really a challenge to try to understand what is going on in these systems, because they're all effectively different ecosystems. Because when we're looking at the microbial populations, you're looking at an ecosystem within an animal ecosystem within the human ecosystem feeding on this plant ecosystem.
Yeah, it keeps going and going. And then even, compost is made from manure of various animals. So it's circular and it's amazing like that.
Yeah, it definitely is.
So what are some of the questions you're trying to answer right now with your research?
Some of the questions that we're trying to answer, I guess the simplest one is, what is a good microbiome? And, you see commercials, like I saw one the other night from Dove on TV where it says, we will protect your microbiome or give you a better microbiome. And I looked at that and I'm like, I was one of the first handful of people to do this kind of research. And I can't tell you even now what a good microbiome is. I can tell you what some really bad ones look like in some cases, but even that's hard to say good, bad. So what we want to do is figure out how we can link a microbial population with end products that we want to track. And people have been doing that. I mean, we've been trying to do that for 20 years. But then to take those intermediate metabolites and link them with animal performance or end products that we release, whether you're talking about methane production out of the animal, carcass quality if we're looking at beef cattle, milk production if you're looking at dairy cattle, carcass quality and amounts in pigs. So those kind of issues are really important to understand. How do we improve our sustainability by improving the precision with which we feed our animals? Because especially when you look at a ruminant, because they evolved to digest cellulosic feedstuffs, when you look at them compared to, say, a chicken, where you're 1.5 pounds of feed for a pound of gain in a chicken, with cattle, you're feeding 6, 7, 8 pounds of feed to get a pound of gain, because the energy density of forage is less and it's a different feed stuff. So that's a really terrible metric in some ways. But the fact is the ruminants traded adaptability for efficiency. And that allows them to utilize these other feedstuffs. And that's really exciting to be able to take these feedstuffs that other things can't utilize and let ruminants like goats and sheep, they can exist in scrub and really terrible environments. And they'll thrive and do just fine in conditions that would kill other animals. And just
so you're trying to find what the cheapest feed, but also the healthiest feed? Or, like it sounds like there's a bunch of parameters that have to be juggled.
Yeah. Oh, yeah, there are a lot of parameters because ultimately, if we're producing food for humans, the animal and farmers in general were charged hundreds of years ago with produce as much food as you can, as cheaply as you can. And we've done a great job at it. If anything, in agriculture, we're a victim of our own success. We've made calories so cheap and protein so cheap that people don't value it as much as they should. And just the miracle of the food supply in the U.S., when you look at it compared to other parts of the world, is just outrageously incredible. But you want to have affordable feedstuffs so that farmers can produce affordable cattle or affordable chicken for consumers to eat who need that protein source, that high quality.
What are some of the parameters that you've modulated, and how has it affected the manure, the types of food, the quality of the meat, for let's say chickens, cows, etc. Like, what are some of the parameters again? What are some of the experiments you've done that proved interesting or had positive results?
Well, a lot of the work that I've done and some others along with us have done have been trying to work on improving carcass quality by understanding and manipulating that microbial population to do things like increasing the amount of marbling, that white fat inside a steak that makes a steak so good. If we can increase that while we're feeding less feed or a even cheaper or feed stuff that can't be used for other purposes, if we can make that happen, we can improve the availability of protein for people and improve that quality of product as well. And that's been exciting to play with some different probiotics that do that. And we've looked at some phytochemicals or plant-based compounds that can do it. But right now, we're focusing a lot on understanding how the genetics of the host animal play a role in driving that microbial population, at least to a certain extent. So we have one of my colleagues here set up several years ago a herd of cattle that he's bred for high feed efficiency and low feed efficiency and high marbling and low marbling. And they've genetically diverged over several generations. So we've got some, what you would call, for lack of a better word, you have some real puke cattle and you've got some really good cattle. And they're out of that same original gene pool. So we can look at those animals' genetics and look at their performance. And we now have the ability here at UGA to measure their feed intake on a minute-by-minute basis, their water intake, how much methane these cattle are producing, what their growth is, and look at all these in a very fine granularity of what effects different feedstuffs have on these animals of very different genetic potential. And then we can correlate the microbial populations that are different with these different outcomes of the animal's physiology.
So what have you seen so far? What are some of the parameters that get modulated now?
Some of the best parameters that change, again, are feed efficiency, how much feed the animals need to gain the weight that we want them to, as well as carcass quality, marbling. So, you can think of it as we can take, in theory, we can't necessarily look at it quite yet and get there, but what her goal is to be able to feed these animals and take them from standard to choice or choice to prime quality steaks, because people like to eat, higher quality steaks. And that's where we're going with it, is how do we produce better quality food for less input and for a reduced carbon footprint?
How much do you think, is there like a theoretical maximum that you could impact, the inputs versus the outputs? Like you said, for cows, it appears to be about 6 or 7 to 1 pounds, 6 or 7 pounds of input for 1 pound of meat. Chickens and other, animals better, but how much do you think you could impact this ratio, for instance?
I think under the right conditions, we could get it to probably 4 or 5 pounds, depending on what we're looking at. But again, remember, we're talking about feeding grass or hay, 4 or 5 pounds of grass or hay versus 4 or 5 pound or versus 1 pound of grain, of corn. So the energy density of that diet, even though you're talking different pounds, is very different. And because we can't degrade and chickens can't degrade, or pigs for that matter, can't degrade the cellulose, then that feed stuff that we're giving to cattle is pretty much useless to other animals. And that's the beauty of it, is they're not competing with anything else for feedstuff. So if we can drop it basically and improve our gain and quality of our feedstuff, then we've really done a lot of good in improving that availability of protein to human consumers. So I don't know that I'm even capable of making a wild guess as to a percentage improvement that we can get, but we can certainly, it looks like we can certainly make some changes.
Okay. I don't know. Are there any novel ideas out there, for how to do this, or have you seen yet the resultant changes in the microbiome and how that's influencing, these properties?
Well, we are literally we're about 2 years into this project where we're looking at it. We've had a couple of students publish papers, and we just are getting our system online where we can measure live animal methane. So this is ongoing processes for us where everybody is trying to get moving forward and look at this holistically for animal production, going all the way from that carcass quality all the way back to the farm into selecting which cattle we're producing. So right now we've seen there's certain bacteria that are highly correlated with marbling. There's certain other bacteria associated with feed efficiency. So we're looking over several years of cattle to try to link these together. And we have a corporate partnership with a company now that they're trying to work on developing diagnostic systems as well as probiotics, where we can look in and see, are these cattle going to be more efficient, where it should be in the farm at the side of a chute, or when you're raising your cattle, where you can check and see, are these the most efficient they can be, or do we need to feed them another probiotic? And if so, what kind of probiotic can we give them? Do they need? So that's where we're trying to go. And we're still in the very early phases. We're trying to figure out how those link together and how closely linked this animal genetics, microbial population, and animal performance and sustainability truly are. Now, on the food safety side, I worked for several years with the USDA, the Agricultural Research Service, and one of my colleagues, and we were on the same team, developed a great product to improve food safety, and that's the development of something called sodium chlorate. And chlorate is it's a chemical. It's made of salt, sodium chloride, and oxygen. And it is a valent state analog. So in other words, chemically, it looks a lot like nitrate. And a lot of gut bacteria, especially salmonellas and E. coli 0157H7, this enterohemorrhagic E. coli that you hear about, the hamburger bug, those organisms are equipped with an enzyme called nitrate reductase, that it takes nitrate to nitrite. Well, this enzyme doesn't recognize nitrate or chlorate being different. So instead of if you provide chlorate, it's going to take chlorate to chlorite inside the bacterial cell. And chlorite is bleach. So that kills the bacterium from poisoning it from inside the cell. And then that chlorate will be broken down by the host animal. So we looked at that, feeding it to cattle, pigs, and chicks, and saw reductions in salmonella, Campylobacter, and enterohemorrhagic E. coli. And it looks like it is a really great product, and it's been going through FDA approval process for a long time now. So hopefully it'll get through sometime in the next few years. But it's pretty exciting to have been part of something that it's the closest thing that I've ever seen to a silver bullet to improving food safety on the farm.
Okay. Well, very good. Where can people find out more about your work? I mean, it sounds really interesting, but it's still in the early stages, I would guess. So where can people find out more?
Well, quite honestly, most of our work is still being published in the scientific journals. We're building a website currently here at UGA for our beef improvement, sustainability improvement team that should be available, but it is available on the website of University of Georgia Department of Animal and Dairy Science.
Okay, any other place, or that's the best place to go?
That's the best place to go.
Okay, all right. Well, very good. Well, thank you so much for coming on the podcast, and I appreciate it. And like I said, it's amazing the cyclical nature of all this, and like I said, microbes everywhere and at every stage. It's very interesting work you're doing. So thank you.
Thank you. I appreciate it.
Before you stop listening, ask yourself, what are 1 or 2 useful things you heard on this podcast that can help you overcome food and fertilizer shortages, skyrocketing prices, the cost of living, or your job being outsourced overseas or eliminated due to automation. Please like and subscribe and tell your friends and family about the Surviving Hard Times podcast. We're all going to need help now and in the near future.